Welcome to the first live show ever of the Immunology Podcast with me, Dr. Jason Goldsmith, and Dr. Brenda Roud, here live at Immunology 2024 in Chicago in McCormick Place. And today we're going to be discussing the role of AI in immunology and what it's kind of means for us now and possibly in the future, and even maybe a little bit outside of immunology as we talk about the larger implications. With us today, we have two people from Seismic Therapeutic. First up is Dr. Jo Viney, who is the CEO and founder of Seismic Therapeutic. She comes to us with 30 years of experience in biotech and has had 13 drugs get into the clinic and 2 be approved, and founded Seismic with the goal of taking machine learning technology and tools that have been used in small molecule and bridging them into biologics therapy. And to do that, she has the help of Ryan Peckner, who comes to Seismic after doing a postdoc at the Broad and has a PhD as well in mathematics, and he is their director of machine learning, computation, and all such things involving AI. And so he is here as well, and we're going to sit here and start talking. So with that, Ryan, I'm going to start with you here. If you can explain a little bit about AI in general, what machine learning is, kind of the basics of what that is, and how we kind of got to now-ish.
Sure, I'd be happy to. Thank you so much for having us here. It's thrilling to be in front of an audience of immunologists. I very much am not an immunologist, so a part of me on that front. But yeah, AI, I think we could define broadly as the use of computer models and algorithms to learn and reason in ways that normally would require human intelligence. And this is a very broad, I think deliberately vague definition, because this field actually, we think of the explosion now, especially the emergence of ChatGPT in late 2022 ushered in a real revolution in how we all use this and interact with it in our daily lives. But this field actually goes back all the way to probably the 30s, one could argue, with the seminal work of Alan Turing, a mathematician who described kind of the basic, logical principles that underlie the idea of an intelligent computer. And he developed the Turing test, which would be, the way to really tell is a computer actually intelligent enough to fool a human sort of adversary. And the field, developed theoretically for quite a long time, the sort of basic mathematical unit underlying deep neural networks, which some of you may have heard about, was actually introduced in the 40s. Some implementation of those ideas was brought about in the 50s and 60s with the first sort of algorithms that could recognize images, and in very simple ways, they could kind of tell whether a black dot was on the left side or right side of a piece of paper, that sort of thing. And then neural networks, this architecture that underlies really all of the major developments in recent years, those were introduced arguably in the 60s with work from Hopfield. So, I just say all of that as a preface to the point that this field is actually quite old. It's not really a new field at all. What's happened very recently, and arguably the last 15, maybe 20 years, that has driven this revolution in AI that we're seeing today is really this explosion in 2 things. One is computing power. So a lot of you have probably heard about NVIDIA, this company that makes GPUs, graphical processing units, which are these core pieces of hardware that enable the rapid mathematical calculations that underlie all these neural networks and other kinds of advanced AI algorithms. And the other is the explosion in data, publicly available data all over the internet, private databases, and also a lot of areas that you might not necessarily think about as being publicly available. These could be sensor data. They could be things like patents and grant applications that are not immediately parsable by a computer. They're not necessarily collated into a central database, but are nonetheless somehow out there. And these 2 pieces were the fundamental factors that prevented AI algorithms in the past from doing what they're able to do today. And when we talk about AI, really what we're usually talking about is actually machine learning, which is the use of large data sets to train algorithms that can generalize, that can make predictions, and that can actually generate new kinds of data resembling what it's seen before, as opposed to other kinds of AI that are not machine learning. One could think about like Deep Blue, the chess playing computer that beat Garry Kasparov in the 90s, which had a lot of ability to kind of reason, but wasn't machine learning per se. So that's my sort of 30,000-foot overview of AI writ large.
We hear a lot about, so you mentioned already with the history, I think that's really useful. And we also hear a lot of terms like generative AI and non-generative AI. We have these large language models. So if I understand correctly, there's a fundamental difference between generative AI and what is non-generative AI. So maybe would you like to introduce a little bit to that? Because then I think it's going to be important for a discussion down the road.
Certainly, yeah. So in the early days, when I say early days now, I mean, say, early 2010s as opposed to back in the 30s. But initially, AI or machine learning began with the goal of making predictions, being able to predict the future, being able to label data that hasn't been annotated in a certain way. So say, you want an algorithm that, given a picture, can tell you if that picture has a cat in it or not. That was some of the seminal kind of problems that initiated a lot of the early AI algorithms. And in the 2010s, those problems were solved pretty quickly with tools like ImageNet and AlexNet that largely came out of Google and other large tech companies who wanted to enable these automated kind of labeling or automated predictions. And we call that kind of AI inferential. So when we make a prediction on a new piece of data, we call that an inference. So this is inferential AI. By the mid 2010s, it was kind of realized that the same architectures that were being used to enable these kinds of inferences were, in a way, learning, more fundamentally, the underlying kinds of concepts that, lead to these predictions. So it wasn't just that, given a picture, it looks for like, okay, here's what an ear should be shaped like, or here's what a nose should be shaped like. And then it kind of tries to do some sort of pattern matching. That could be one way you could look for a cat. Instead, these algorithms were learning about what a cat looks like in a more fundamental way, encoded deep in these so-called latent representations that are the core of these neural networks that perform these predictions. And it was realized that because of this sort of embedding or encoding of information within these models, you could actually generate new instances of cats. By kind of moving around those latent representations of these ideas, by kind of manipulating, or hallucinating within this sort of deep within the layers of these neural networks, you're able to generate new instances. So you can now turn to a computer and say, I want a new cat picture that no one has ever seen before, or I want a cat on a unicycle on Mars playing a bugle. And these algorithms can do that, and that's the era of generative AI, which interestingly was not really part of the initial genesis of AI. It really began with the goal of prediction, and it was a fairly late development, the realization that these latent representations could lead to generation of new data.
Were people surprised? Were not expecting that?
I think they weren't expecting it to happen so quickly or to be so successful. Is one point. The other point, I don't think they were expecting it to be enabled by these, kind of very human interpretable, text interfaces where you can, without any programming experience, go to DALL-E or ChatGPT and just put in a natural language prompt and get back what you want. I think that happened much more quickly than was expected.
Would it be correct in saying that generative AI was assumed to be more the realm of general artificial intelligence, which was not going to be from these machine learning models, and then, oops, we got generative AI a little early in the cycle?
I think that's a really interesting question, and it could be a little more philosophical. I would argue that generative AI is still machine learning simply because it still needs a tremendous amount of data in order to perform that kind of novel generation. AI that was truly maybe general or not machine learning would mean AI that could reason with no data or a very scarce amount of data, which incidentally humans can do. Essentially, given one new instance of a cat, often someone or something someone hasn't seen before, they'll be able to generate something that kind of looks like that, but maybe has some differences. And yeah, the AI is really, or the machine learning, these generative models aren't there yet.
So now going to seismic a little bit, Joe. In the intro, I talked a little bit about AI for drug discovery, molecular dock, small molecule, and you guys are really taking that into the biologic space. So could you talk about that a little bit from what you guys are doing for drug discovery and biologics, maybe even weaving in a little of the old AlphaFold tale, which some of us may have experienced at some point in our lives?
Yeah, I'd be happy to do that. So as was introduced, I'm an old person. Thank you very much, Jason, for highlighting how many years I've been doing this. But I'm a long-time drug hunter, drug discoverer. I've worked in biotech for most of my career. And I've always worked in the autoimmune disease space. I've worked across a number of different modalities, but in recent years, I've really focused on protein therapeutic drug discovery. So in 2021, when machine learning and the application to small molecule drug discovery was fairly well established, myself and my co-founders of Seismic were really interested in thinking about, could we apply machine learning to protein therapeutic drug discovery? So it's a little bit more complicated, needed some additional data sets, needed some additional algorithms to be invented, I guess. But we were excited by this prospect. So Seismic was founded with Tim Springer, who's a longtime structural biologist. He had been collaborating with a professor of machine learning at Harvard, Professor Debbie Marks, and they had this idea of like mixing the two together. So Debbie had a pretty good history of using machine learning to measure or evaluate or predict protein fitness, but actually hadn't done much in terms of experimentally testing it. So that was really how I came in, was can we weave all of this together and can we think about applying it, and can we actually make protein therapeutics that are better? Can we make them more quickly and more efficiently and have all the drug-like properties that make them useful for treating chronic autoimmune disease? So that was the start of the company. Obviously, I needed colleagues like Ryan, who have a background in machine learning, to help us do that. I sort of brought the drug discovery stuff. But what we've seen since 2021, when we thought this was a bit of a far out idea, is actually a plethora of new machine learning in protein therapeutics drug discovery companies. And actually all of the large companies, large biotech and pharma, I think all now have machine learning and AI groups within their domains to help advance and accelerate proteins. This is great news for patients. So sort of now narrowing down onto Seismic, I was a little bit skeptical in 2021, and so didn't want to put all my eggs in one basket. So we decided to work on two different classes of proteins. So one of them was antibodies, and one of them was enzymes. Now, the machine learning application to enzymes was much better known, but the application of antibodies for treating chronic autoimmune disease was also much better known. So they were a little bit non-overlapping. What has happened since then, as Ryan mentioned, was ChatGPT came out. So I think everyone knows that. You mentioned AlphaFold. This has been an amazing contribution, not just AlphaFold and Google being involved in this, but David Baker coming out with so many new algorithms that I'm sure Ryan will talk about some of these new ways where we can actually predict protein structure based on sequence. And now we can actually think about how do two proteins interact, or as has now been more recently, we're involved in this, many other companies are involved. Can you actually look at the structure of a protein and the sequence of an antibody, and can you in silico predict where that antibody is going to bind? And that sounds really straightforward until you actually start thinking about the three-dimensional aspects of this, because antibodies don't bind linear epitopes. They bind the surface structure of a protein. So you really do need machine learning to help you visualize that. I think, as Ryan said earlier, it's the machines are doing what humans sort of could do, but doing it much more quickly. So that's the bit that really shortens for us that design test cycle time. I don't know, Ryan, did you want to talk about some of the algorithms that have really helped you, some of the algorithms in the public domain, and then some of the algorithms that you've built?
So as Joe mentioned, we have these two domains that we've worked in, enzymes and antibodies. And we've actually, initially the techniques we were using to optimize either or design either were very disparate. When you're dealing with an enzyme, we we're taking this approach of so-called evolutionary sequence models, where generally these enzymes have a lot of evolutionary homologs, often from similar species. If you have like a bacterial protease, which is the first case we were looking at, there are thousands of bacterial cysteine proteases that you can look at and sort of train machine learning models to learn what are nature's rules of what it is that makes a bacterial protease. And in that way, you can take a given starting sequence and kind of optimize it. You can supercharge it with these algorithms that have learned what it is that makes it functional, what it is that makes it solubly expressed and stable, and all these properties that we like that are favorable for developability and optimization of function. On the other hand, for antibodies, you don't have that luxury because CDRs, particularly the heavy CDR3, of course, doesn't have an evolutionary history. It's often unique to that specific individual, and therefore you can't use these models. And so we were using rather different techniques, these so-called protein large language models, where instead of trying to learn properties that are very specific to that antibody or antibody family, as we're able to do with, say, cysteine proteases, we're learning much more general principles, models that are trained on hundreds of millions of protein sequences, these so-called universal protein language models that say, okay, in general, what are the properties of a protein sequence that allow it to fold, to express well, to be stable? We don't want like 5 prolines in a row or something like that. These sort of, there's some heuristic principles that we as humans could come up with, and that's how this field initially started with antibody optimization, rational design, a lot of heuristic principles, and I say that not disparagingly. A huge amount of expertise that ultimately fed into the design of these machine learning models.
Don't disparage the protein engineers.
Never.
That's part of your team.
No, very much working closely with them to design models that incorporate that kind of expertise, but also can learn on these immense data sets to get a very general picture. And what we found is that with those kinds of models, we're able to optimize antibodies, again, to do things like not just affinity maturation, but affinity maturation plus, deimmunization, affinity maturation plus reducing aggregation potential, viscosity, all these other properties that we care about. So basically enabling multi-objective optimization through models that understand the fundamental tenets of what makes a good protein.
So I actually like that you mentioned that, because that, from my experience looking into what the research is doing, I see that pattern a lot, that first the algorithm needs to learn the language, like how the whole spectra looks like, and then you can kind of train it again on something that's closer to what your question is. I've seen that also done for promoter sequences, for DNA analyses, and I find that on the one hand it makes sense, because I think as humans we cannot do the same too, right? We first want to feel comfortable with the space, and then if we know a little bit more than what our problem is, that gives us kind of this intangible understanding. And I find that it's kind of eerie that these models seem to do the same. But I guess there's a general pattern in this field, right?
Yeah, and there are actual formal principles that encapsulate exactly what you're describing. So there's the whole field of sort of transfer learning, where you would, if you want to develop an improved antibody or something, you could first take a model that's been trained on all protein sequences and then refine it just to antibodies. You could even take a model that's been trained on just human language and then refine it to proteins and then refine it to antibodies. And that has been done, especially these so-called, people may have heard of BERT models, BERT, which stands for bidirectional encoding representations from transformers, which are very general language models that then folks have applied, this ProBERT, there's anti-BERTA, these models that have been refined to the specific task of interest, in a formal mathematical way, but it's doing exactly what you're talking about intuitively.
And maybe I can take it back to the reality of immunology and away from theoretical math for this audience. So one of the so in plain language, if you think about the area that Ryan first mentioned, natural proteases, there's 3,000 or so proteases. So Ryan's machine learning team sort of integrated all of that data. But how he now interacts with our protein engineers is the protein engineer says, I need to make one single amino acid change here to help with stability of the protease I'm trying to make. But often in traditional protein engineering campaigns, you make that amino acid change and the protein doesn't express, it collapses, it loses its function. And so the protein engineer is like, well, that's great. So let me try a few different other amino acid substitutions there. And maybe it leads to something, or maybe it doesn't. But with machine learning, now the protein engineer can say, I need to change that amino acid here, but the protein doesn't express. So what exists in those 3,000 other proteases three-dimensionally, the epistasis, the relationship between these other amino acids. And if I want to make a change here from one amino acid to another amino acid, what 2 or 3 or 4 other amino acid changes might I make? Because I can look at these 3,000 proteases and see what they've done in nature. Now I can include all 3, 4, 5 of those amino acid changes, and my protein expresses. So that's the beauty of having machine learning. You still need the protein engineers at the front end, but now instead of taking, I don't know how many iterations of mutations and trying to express and measure function, you can actually really reduce the number of rounds of engineering that you do, and you can incorporate multiple different amino acid changes in a single round of engineering, and you end up having a molecule that you can express. It's more stable and still has the functional activity. So that's, I think, what's really exciting about the application of machine learning in protein engineering, especially in the immunology space.
So I wanted to go to the generative AI portion of this. So you guys are at the point now where to take, like, you had your protein engineer, I want that amino acid change. You go to the point where, like, hey, I have a protease. We'll just keep on that. I have my first round, I had some off target, right? Those that don't know off target, that's the side effects you don't want from your protease on something else. I wanted to really bind this. Can you, robot, I always call AIs robot overlords. Robot overlord, can you help me improve the binding for this target, but not for this sequence here? I want that binding to be way worse. Can you help me do that, AI? Are you guys getting to the point where you can prompt the system to do that double push? You're not saying which amino acids, just like, I want it to bind this one more and this thing less.
Yeah, so this is truly science fiction drug discovery. So I think that it is possible to use AI to create molecules. I think how those molecules will perform and be useful of drugs, we've still got a little bit of a ways to go. But I can't remember, what's the, you've got a protease where I don't remember, like big chunk of it is sort of generated fictionally.
Yeah, we have done actually exactly what you're describing, where we modulated, we had a protease that kind of was hitting a couple of different targets, and then we used our algorithms to re-engineer it to be specific to only one of those. Now the caveat is, it wasn't, as you're saying, just, sort of go to the computer, say, I have this problem, can you re-engineer it, do this thing. We still needed a huge amount of protein engineering and rational design expertise, and actually that particular case relied on having our, wonderful structural biology colleagues, solving these different structures of, like, the protease and complex with different targets, sort of identifying here maybe the sort of general drivers of the non-specificity. Then we were able to go and apply the AI and say, okay, well, here, now redesign the sequence to make it only specific for specifically one of those targets. And with fewer than a plate of designs that came out of that AI, we were able to get a high affinity, highly specific protease variant. But again, we're not at the point. I think it's very fair to say we're not at the point where this field is replacing any of that expertise. It's really like the terminology they use at Microsoft. It's really a co-pilot. It's really assisting and massively accelerating that process.
Is part of the reason we're not there yet because there's a lot more cat pictures, to go back to that analogy, than there are protein structures, and are just the knowledge base is so different that while I can tell Midjourney to draw a cat in space playing a bugle, it's different with protein structure prediction.
So structure, I actually think we are very close to a point of fair journey-like generative capacity, and I can talk more about that in a moment. But I think here the fundamental element isn't structure but function, and a database that describes specificities of proteases. There is such a database like MureOps, but it only has something like 4,000 entries. With annotated function, a lot of proteases, as everyone knows, are extremely non-specific. You can't really learn much from them. So you don't have a lot to go off of when it comes to function, when it comes to context, biological context in that way. For structure, I think we actually, putting my head above the parapet, I would say we are there. Very recent methods that came out in 2022, 2023 employed this stable diffusion or denoising diffusion idea, which is the same mathematical technique that underlies these, DALL-E and Fair Journey, generative image generators, where you sort of, but they're applying it to proteins, where you sort of begin with a protein structure and you train the models such that, as you start jiggling that structure, you start adding noise and sort of turning it into just a total mess in a sequence of noising steps. You train the model to denoise, so to reverse that process, so that if you began with a clump of noise, the model would learn how to sort of assemble those atoms into a real protein structure. David Baker's lab at University of Washington, Seattle, as Joe mentioned, have been pioneers on this front with the RF diffusion method. Also Generate Biomedicines, a startup also in Somerville. They've applied this, developed a method they call Chroma to generate, brand new proteins. So new structures that look nothing like anything that we've seen in the PDB. And that really all relies on the explosion of structural data over the last, 30 years, where we now have hundreds of thousands of structures, through huge hard-won efforts from crystallographers, cryo-EM, all those different fields. But I would say we are there when it comes to structure alone, but we're still a long way to go when it comes to function.
So trying to look a bit more in the big picture, I attended your talk, Joe, and you were, for example, using, for more immunological research, you were using machine learning to help you predict which antibody would bind. In this case, you were looking for antibodies that were agonists to PD-1, and you used that to say, well, this antibody is probably going to be better at being an agonist and not interfering with the normal functioning of these receptors. So I see this also as an application in the immunology field, that we can start really much more rationally, much more quicker, designing better protein structures for various processes. In this case, you had very good results by using this to find the right antibody for your purpose. But let's take a little bit of a step back, trying to maybe prioritize those uses that maybe our listeners might be more interested in. What else is there in it for AI to do for us? So we have protein structure prediction, but if you look at the literature today, and I think that's also, you start reading papers and you see that people are actually starting to apply analyzing genomic data, analyzing cellular signaling, maybe a little bit further down the spectrum, looking into patient records and trying to predict diagnosis or treatment options, things like that. So from your perspective, what are the other notable applications of AI that people are using now, or are we probably going to start using, anytime soon?
Yeah, there's many, and actually we've sort of really focused what we've been talking about today on the actual engineering and making of a therapeutic. But you're absolutely right. If you go back to the beginning of any drug discovery process, you really have to think about what cells do you want to target, what pathways do you want to target. There's been a big emphasis on using AI to help us with target identification and prediction, really sort of tying what targets are misregulated in what diseases, and really giving you that sort of starting point of where you want to be. That does require really well curated data sets specifically. I think that's been one of the challenges, and for companies that are really focused on using AI to help with new target identification or new pathway identification, having unique data sets is really, really important. And having large data sets is really important. And the reason, and when I say curation, I mean, if you're looking at patient samples, you can't just say, oh, I want to look at a patient with inflammatory bowel disease, or I'm going to look at 2,000 patients with inflammatory bowel disease, because those patients are not all identical. For example, some could have ulcerative colitis and some could have Crohn's disease. And you say, okay, well, I'm actually going to focus just on the ulcerative colitis patients. Well, there's many different stages of inflammation. It's a relapsing remitting disease. There's many medications that those patients are on. And so really curating those samples and having that built into the AI platform is really important. So that's sort of the very front end. You've then mentioned around, well, you've already got the pathways, the targets. Now, what about the signaling pathways? How can we use AI for that? That's very similar. I think that's much more in our labs. That's in our hands as immunologists, really trying to think about the interconnectedness of the many different pathways. These are not, we often think, oh, I'm going to work on a BTK inhibitor because that'll inhibit B cells. Well, BTK downstream signaling overlaps with many other downstream signaling pathways. So that's really important to use AI to sort of desegregate that. Then you can say, well, now I've got my target. Like us, we're using ML for protein therapeutics. I mentioned that ML has been used for small molecule therapeutics for a long time, so we've sort of covered that bit. But you've now also mentioned, how can we use it afterwards? How can we comb through the plethora of data that already exists in patient records? How can we actually now say, can we predict which patients are likely to respond or not respond? And can we use that information from clinical trial data to help us stratify patients up front? So that's really important. And then I want to reverse a little bit from that and say, part of the drug discovery process that we often don't talk about is that, year to 18 months of testing molecules to make sure that they're safe, they have efficacy, they're not going to cause harm. And there's a big effort in AI and ML right now to see, can we minimize that time? Can we reduce the number of animals that we have to use to look at the pharmacokinetic, pharmacodynamic, and toxicological profile of these molecules before they get into patients? Because that bit's really hard to shorten, because we can't take anything into humans that hasn't been very well tested. So that's an area. I know there's companies that are focusing on that, and I think that's going to really help revolutionize how quickly we can get medicines to patients.
I wanted to jump on the tox bandwagon here, being married to a toxicologist who doesn't listen to the podcast because it makes her uncomfortable to hear my voice, so I can talk about her as much as I want. That being said, you mentioned this point of like, that's a really big barrier, is there's the time it takes, and then you often not mouse, right? It's monkey studies, and they're expensive, and then there's the harm to animals. FDA has started to be comfortable with machine learning for like histology prediction or some other algorithms in medicine. As someone working on AI on another part, but also having been in drug discovery and thus been through many an IND in your life, what do you think it's going to take to get regulators to be comfortable with, here is, 2 studies, not 10, and a bunch of machine learning AI stuff that says it's really safe. I promise the robots aren't lying.
Yeah, I think the onus is on us to demonstrate the translatability of using machine learning for that predictive analysis. So it is scary for many people to think about AI and machine learning. And I think when things are scary and sort of look like a black box, it's really hard to understand, how can I actually use it? And I think the regulatory agencies have a very important job of making sure that we do no harm. And so it is going to take a little while for us to close that gap. I don't know how long it's going to take for the regulatory authorities to be comfortable with 1 or 2 studies versus many studies. But I think that's, as all of us who work in the drug discovery industry, I think that we're going to have to strive towards that, because there's got to be better ways to do it. And there's got to be quicker ways to do it, because every day that you delay getting to a patient, particularly for some of the diseases with high rates of mortality, and I know, Brenda, you work in the oncology space, this is really important to sort of have therapies that are going to be beneficial in patients as quickly as you can. Or infectious disease. I don't know if anyone here works in infectious disease, that I think the COVID pandemic taught us that actually, every day really does count. We need to get, vaccines to people. So I don't have the answer to that. I'm glad I'm not one of the regulatory authorities who's having to make that decision, but that may be because I've sort of had most of my career in the creation phase rather than the regulation phase, of the regulatory phase of drug discovery.
I think, personally, and I don't know anything about toxicology, so take this with a grain of salt, but, I think one major difference is the cost and the time it takes to generate a toxicology profile versus histology, where you have, millions of images that you can spit out with, all these cell imaging techniques, which have led to these state-of-the-art, image recognition algorithms for histology. So in a way, it's not surprising that it's not that the algorithms are better in histology per se. It's just that there's so much more data and higher quality labeled data versus toxicology. I imagine there's much less, you don't have a bunch of, say, variations of a given molecule that are hitting this, where you're looking at the tox profile. You probably have one, maybe two. So you can't say like this one did well and this variation of the sequence did poorly. And then so it's much harder to train models just because the data is much more sparse.
I want to come maybe back to that. First off, how do you get the data? So as a company or as a researcher, how do we manage to get this data to train these algorithms? What is the future of getting this data? I'm thinking of, if you think of ChatGPT or the more kind of popular language models, number one, apparently they're running out of data. I learned recently that they have literally gone through the whole internet already, and that's it. And then now they don't know how to keep improving them. And on the other hand, the source of that data can be problematic. I mean, copyright, sometimes the data is skewing the algorithm in a way that then you end up having a final product that is not what you were hoping. So on the one hand is what comes in. So as Jason has said, garbage in, garbage out. So you need to be very careful about that. So where are you getting? And how do we either solve the problem of the black box or become comfortable with the idea that we don't know what's going on in there? Or can we retro-engineer in a way what's going on in order to understand where those predictions, where those results are coming from, if we can? What do you think?
Yeah, I think there's a tremendous amount of that can be done already with publicly available data if we allow ourselves to be creative in thinking about how to use it, what kinds of models we train. One of the huge advantages of machine learning, as opposed to more classical statistics techniques like regression, or even thinking about like more classical machine learning algorithms like random forests as opposed to neural networks, is that neural networks in particular can ingest a huge amount of very diverse data modalities and integrate that all into one single kind of oracle that can take in be it text data, be it omics data. I mean, a really striking recent example of this was from in silico medicine in Hong Kong, where they have now the first end-to-end AI-designed small molecule in clinical trials and entered phase 2 recently. And this is for idiopathic pulmonary fibrosis, where they fed in, they trained a model that was trained on multi-omics data from public databases, genomics. They looked at patents, they looked at clinical trial data, all these different sources of information that were combined to identify, a rich potential source of novel targets in IPF. And they found this TNIC as a potential target. And then what they did, addressing the second part of your question, is all this so-called transparency analysis, where you look at, okay, so the AI has spat out this TNIC protein, but the AI doesn't know why. It just knows that there seems to be a lot of correlation. And they actually looked at, if we do our own kind of very human interpretable pathway analyses, looking at correlations with these omics data, we see a lot of evidence that suggests that what the AI is giving us is actually a really valid idea. And then from there, you go and you do your assay work and validate the idea that this is a valuable target. And I think that's the huge advantage that we'll always have in this drug development context, as opposed to an enterprise kind of software like ChatGPT. We're always going to be validating the predictions that come out. And as we generate that data, more to your question, we can feed that back into the model to get the model to give us improved predictions. You could say, actually this thing that you suggested is a poor target. Now incorporate that into, back into the training. So the fact that you're beginning with something very large, doesn't necessarily mean that you're restricted to using public data. There's always a sort of cycle in that way.
I think one of the challenges is how do we share data between enterprises? I think that's probably where you were getting to. And I think that I don't have an answer to that. But if you think about the wealth of data that exists within the various companies that exist in this industry, there's a ton of information that I think could really be helpful. But, it's we don't have a way to access all of that yet. I think you were mentioning like, oh, if I look at a paper and I want to access the data set, how can I do my analysis on that data set? And you can, but that doesn't necessarily allow you to integrate that data set with another analysis that you do from another paper. So I don't think we're there yet, but I do think that the feeding back of your own data into your original data training set can be helpful, but it would be amazing if we could do that sort of more globally.
I think you talked about an interesting way of working. So we have a crowd here. How many people in the audience have spent 3 or 4 months, just by a raise of hands, like, or even 2, reading a bunch of papers to figure out what their project could be in grad school or postdoc or anywhere in industry, right? Like, right, exactly, almost everyone. So it sounds like what we could do, we're talking about ways of working with AI, is you could come up with a hypothesis or 2, feed it to the AI, and it can, air quotes here, read the papers for you, so to speak, and tell you which of your 5 ideas may be worth you then reading the paper, doing some preliminary work, and going from there. Is that a way of describing that co-piloting experience, where instead of trudging in the library for 6 months, you do that second, not first?
Oh, absolutely. I think people are doing that already, both in academia and in industry. There's the in silico example I mentioned, and then there are commercial software, commercial efforts to bring that exact kind of high-level sort of hypothesis generation capability into clinical context. I know there's a company called Insight that does this sort of thing, ingesting huge numbers of papers to give you a Google-like interface where you can say, okay, what would be an interesting target to go after in this like epithelial disorder or whatever, and it can reason with you in that way, and then say, here are all the papers I read that were relevant, here are the main points, and let you go back and really do that kind of human interpretation.
Yeah, but be careful though, because it's not always right.
Oh, certainly. Yeah, that's why
I don't know,
buyer beware.
Yeah. So have you ever, so I asked, so my husband uses ChatGPT to like summarize a lot of the documents that he's doing. He works in the nonprofit world, and he said, mostly it's right. And I said, well, I don't really know how to do that for science. So I asked some science questions, and, ChatGPT was kind of mostly right. And I said, okay, here's the perfect test. So I said, ChatGPT, what is Joe Viney famous for? Have you ever done that? Has anyone in the audience ever asked ChatGPT what you're famous for? Well, it was mostly right. It was really scary. And there were some things I'd kind of forgotten that I'd done in my career. So that was a plus. And then there was these other things that I'm like, I really don't remember doing that. And actually, I really had never done that. So buyer be warned. I think that, one of the things you're hearing Ryan and myself say is that these tools are all really great. They still need some human oversight and some logic to put in, some logic applied. And that's hard when you're in this business where we're all exploring new frontiers. We have hypotheses. We don't actually have the answers. And so whilst it might be good in grad school to have ChatGPT figure out what your project should be, just be careful that you, pressure test that it's accurate before you go off and spend the next 3 years following a path that's not a good one.
I guess that the whole hallucination issue is, especially in these language models, I've also once it's like, oh, maybe I can just, what do you know about T-Rex in transplantation? And it's like, oh, these things, and can I have some citations, please, so I can expand? And half of them were completely made up. And many of us have already tried that and seen that. But it is good to know that there are some portals or some applications that are trying to put that a bit more into context and generating specialized AI packages, so to say, to solve this issue.
Actually, just to pivot off of that, I don't think this is any different than being down the pub and having conversations with your lab mates, right? Some of your lab mates have got good ideas, and some of your lab mates maybe don't.
Maybe they're hallucinating too, if they've had enough to drink.
And so, it's just another, it's just another tool. And as scientists, where we are all trying to think of something new that hasn't been done before, I think any of these prompts are good. And even if some of the prompts are red herrings, it makes you think about it.
Is there other kind of future of AI that excites you, or that you think it will come or should come? Maybe Also, in the broader sense that we are about to experience, but it's not there yet. Something
You mean like broadly or
broadly,
in
like in
science
general.
specifically? Well, I think that the era that is going to be coming, which is a little scary, even for me, but right now these AIs are kind of the brain, but they're not hooked up to the body yet. So having embodied agents, where you have an AI, be it encoded into a robot, be it encoded into some kind of hardware, where it can actually interact with the physical world, I think that very much is going to be the next frontier. Of course, much easier said than done. I mean, the basic case of this, which has been around for a very long time now, are self-driving cars, which we all know, the joke is self-driving cars are always 5 years away and never quite here. But I think there are simpler cases where it doesn't have to navigate that kind of complexity. A very relevant case, for this audience, this conference, would be like lab robots. I mean, imagine how great it would be. Already having pipetting robots saves people a huge amount of time. Now imagine, a series of pipetting robots where you could just tell it in natural language, here's the experiment I want to do. Maybe even tell it to order the reagents for you. Maybe they come in and, automate away a huge amount of that, kind of tedious, repetitive work and let you really focus on the creative, fun stuff. So I think the physical agency is going to be the next frontier.
Are you telling that robots are coming for our jobs? Is that it?
just for the parts of your job you don't want to do yourself.
The boring parts.
The boring
I think,
parts, yeah.
It's I still have a repetitive stress injury from washing my own manual ELISA plates, my ELISA plates manually, and pouring my own sequencing gels, and I know that we were all scared about our jobs would go away and what would bench scientists be doing, but actually we are now have more time to be more creative. So, but actually, I want to check: do robots need to wear PPE?
I don't.
So continuing on the prognostication then. So we talked about, embodied AI. How about the big scary one, the general AI that is self-aware and doesn't just fake humans out on the Turing test, but is aware? You think that's the old joke, 5 years away? It's more like, oh, it's always 20 years away. But do you think we're now at the point where it's not always 20 years away?
I think 20 years is more realistic, maybe even as a minimum. I think there's accumulating evidence that large language models, which are the dominant mode of AI, right now, really aren't capable of AGI, artificial general intelligence. They are not self-aware. They don't know what they're saying. They're just spitting out, these words that seem to complete a sentence in a plausible way based on data they've seen in the past. So we call them stochastic parrots for that reason. And we've seen, as Brenda referred to, they're starting to run out of pertinent data. They are hallucinating. They're getting worse at certain tasks, which is surprising. Actually, in mathematics, where you would expect them to get better, they've been getting worse over time. And moreover, it used to be thought that there were these scaling laws that said that with just a tiny bit more data, these models would get exponentially better. Actually, we're seeing the reverse, that we need exponentially more data to get a linear improvement in the performance of these models once they hit a certain wall in terms of the training data they've ingested. So I think there needs to be a fundamentally new kind of approach to the sorts of models, the sorts of paradigms we use to train them. And if you're interested in that field, Gary Marcus is an AI researcher who has a lot of, I think, really interesting ideas and is a nice voice of sanity against some of the AGI hype that you hear coming out.
I don't know. Do we want self-aware like scientist robots? What they're gonna fall with anxiety and imposter syndrome and the whole thing? I think they're better off just being like good, smart pipetters. I think probably even the best for them, to be honest. No, really, it's so weird that we're talking about these things, isn't it? It's like I'm always kind of baffled by what seems to be the world we live in now.
Right. Well, I guess I would point out though that as a bit of a counterweight, AI and machine learning have been in science, in immunology in particular, for I'd say arguably 20, 30 years now. Maybe not in a way that's as visible as something like ChatGPT. But if you go back to the early days of like T-cell epitope prediction, for example, to look at something very immunology specific, the first machine learning algorithm for T-cell epitope prediction based on training on data of known peptide MHC affinities came out in the early 90s. It wasn't great, but it was there. it wasn't until the early 2000s when you have these large databases of peptide MHC affinities, in particular IEDB, the immune epitope database, that folks are able to train really very accurate neural networks to enable that kind of prediction. So the NetMHC is now the leading tool in the field. I believe came out around 2005. There are also tools for B cell epitope prediction, new tools for predicting immunogenicity, looking at antibody, trying to design paratopes. All of this has been going on, continuously. It's just now, this underlying continuous process. And then there are these moments when things kind of surface up in a really killer app or killer sort of tool like ChatGPT, and suddenly people pay attention. But if you look, it's there. It's been there for quite a while. So it's much less scary once you realize that it's been around for a long time.
I always remind people that Google Maps is machine learning. It's been around for a long time. So I think the point is it's been out there for a long time in one way, shape, or form. And even a calculator is machine learning to a certain, that's the first thing that was doing for us. We all enjoy those now. So I think it's time for the studio audience to get involved here.
I know you talked about the protein for drug development and as a vaccinologist. So what AI can do for vaccines? For instance, we know an example of spike protein to develop antibodies. Can AI be used, for instance, to predict B cell epitopes for ADCC, for instance?
And it has. So in fact, for spike protein specifically, Generate used AI to develop a lead antibody. It only took a matter of days once they had the structure of spike. I think it took them less than 2 weeks to design antibody sequences using AI that would target conserved epitopes that were resistant to mutations in the virus. And that led to now their clinical lead program for COVID. So, yeah.
Yeah, so I think, and that's actually a really good sort of segue. One of the things we and others have been working on is actually masking those B cell epitopes so that you remove immunogenicity from proteins. So as a vaccinologist, you're trying to find the most immunogenic epitopes that can drive immunity so that the vaccine will work, so you can eliminate the infection. And for those of us working in the autoimmune disease space, one of the challenges with protein therapeutics is sometimes you don't really know till you get into the clinic whether that molecule has any immunogenicity. So building algorithms that can predict the T cell epitopes that drive T cell recognition, which then give B cell help, which make antibodies, or looking at the cells, the surface of proteins to remove regions that can be known to be immunogenic is really helpful in our space. So that exists. We're using it in 2 different ways already in molecules that are getting into patients.
Hi, I think one sentence that strikes me the most is structure to function. So in the future, so how do you envision that we close this gap? And I envision that to be able to make this correlation, we need high quality, large quantity functional data sets, which will be considered pretty challenging currently with our in vitro functioning. And we always talk about physiological relevant in vitro assays in the industry. I'm curious to hear your thoughts on the future of this gap.
Yeah, maybe I can start a little bit. So I think one of the best ways to predict whether something you make is going to have function is to learn from nature, because nature has already built in and evolved to have a functional activity. So when we look at structure-function relationships, if you learn from nature, you can really start getting a heads up. Brenda was just talking about the presentation I gave earlier today, looking at PD-1 agonists. One of the things we were really interested in doing is mimicking the features of the natural ligand for PD-1 agonism. So that's one way that you can really close that gap. Instead of just coming up with things that bind to PD-1 that may or may not agonize, if you learn from what the natural ligand does, maybe you can short circuit that and actually get there a little bit more quickly. So it does, so that's good for known function. I think if you're asking about structure function relationships where you're trying to have a new function that hasn't been thought of before and doesn't exist in nature. I don't really know. Is there a really shortcut way, or is that still going to be experimental testing?
No, I think the shortcut there would be human expertise. So like the example I referred to previously where we engineered protease function, brand new function that hasn't been seen before. And that came by integrating protein engineering and structural biology expertise with the AI. And that's how you can leapfrog the need for a huge amount of data. And then I think, and there have been examples like the Baker Lab had a case de novo design of luciferases with novel substrate specificity. And there it was this integrated, sort of moving between the human expertise, the AI's sort of, structural design of novel scaffolds that would be able to engage this new substrate and that kind of back and forth. And, until we have these huge functional data sets, which I agree are probably many years away, I think that's going to be the way to go. And it also helps with that issue of interpretability and the black box if you have human experts involved the whole way.
Okay, I have a much less technical question. I've been in conversations a lot lately talking to graduate students and people early in their careers about AI, and that we all think that by the time we're later in our careers, it's going to be really integral and we're going to need to know a lot about it. And it might not be that someone's an expert in AI, but that everyone has some baseline knowledge. So kind of wanted to see if you guys agree with that statement. And along with that, two people earlier in their career who think they're going to need to use AI, how do you recommend they get into that in the beginning? Like, are there resources that you personally really like? Because I think for a lot of people right now, it's really intimidating, but they're seeing the progress it's making.
Yeah, don't be intimidated, because you just need to work with people who know how to do the machine learning and AI. I mean, I'll say, we all have iPhones. Do you know how your iPhone works? This morning I woke up and I had a black screen. Immediate panic. Had no idea. I didn't actually need to know how my iPhone worked, but I did need to know how to use my computer to look up how to reset my iPhone. So that I use as an example of, you don't need to know how to create things through AI, but I think you need to understand what the potential utility of AI is and know how to use the tools that have been built by people who can do machine learning. So this is as a non-AI, non-ML expert. I know the power of what it can do. And I think the advantage that people have who are not experts is that actually you can ask people who are the real experts, can you do this? Can you do that? Can you do the other? And that's one of the conversations that we often have around the coffee machine at work is, hey, Ryan, can your team do this? Can your team do that? And Ryan's like, oh, not another question that is, far-fetched. But every now and again, it's like, well, actually, let me think about that. We might be able to go away and do that. So it's really, it's really important as you think about creating new approaches, finding, new pathways in immunology is just have dialogue with people. And we all have different areas of expertise. We've all sort of, some of us are intracellular signaling pathway people, some of us are soluble mediator pathway. You have to know enough to ask the questions, but you don't have to be the expert in everything. So that's my view, but I don't know, Ryan, do you have a different idea?
Oh, I completely agree. And to your two questions, the first is yes, I think everyone should be aware of the capabilities because it's a very powerful tool and you deserve to have that tool in your arsenal. And you're kind of tying one hand behind your back if you don't let yourself employ these technologies, many of which are free and open for anyone to use. To your second question, I would say yes, definitely collaborate. But even if you don't know anyone who works in machine learning, I would almost certainly there's something in your own work, a problem you yourself are encountering that you could apply AI to in some way. And so I would definitely encourage everyone to look at something specific you're dealing with, whether it's looking for a gene that's a potential target or a potential biomarker, or even trying to analyze some of this mouse data in a certain way. Start to think about, is there a way you could apply, you could answer that question using AI via making novel predictions about data that hasn't been seen before. And from there are a lot of basic tutorials you can find online, a lot of basic tools that will get you the basic flavor of what that is like without having to become an expert or get a degree or something along those lines.
To add to that, I'm actually a really big fan of EDX, has a bunch of wonderful courses, some of which are free or $50 that get you a pretty good understanding of a lot of things, including AI, or even just taking a basic course in R, that type of things, that you get used to the notion of, hey, I need structured data, because you keep hearing data and structure and annotation. I kind of need to know what shape of the object I need to hand to the AI is, and that's something you just get by practicing and doing and finding a problem and solving. And it can be in your regular life too. It doesn't have to be work. You can use AI to help you with something at home, a craft project or something.
You have to have an understanding of the output, and that if you have enough understanding of the tools to get you the output, that's what we all should be doing.
And a little of the input, like I know I got to do A and B, and,
Yep.
in the middle you don't have to be an expert.
Yep.
Yeah, just to clarify, EDX, is that a massive online?
Yeah, if you just EDX, it's
EDX.org.
yeah, edx.org.
Or Coursera. Also.
Coursera is similar as well. Yeah.
So, more questions?
Yeah. Hi. So I'm in high school right now, and I'm researching using AI to actually filter out some single nucleotide polymorphisms that are found in a breast cancer cohort. And so basically, what I'm using AI for is just these like statistical problems. Oh, hey, we've got a bunch of single nucleotide polymorphisms. Which ones are relevant to our project here? But I was wondering if you guys are excited about any future uses for AI in developing mathematical modeling, maybe using geometry or anything like that, modeling to prevent or understand cancer better.
Yeah, definitely. Cancer has been, I think, a major focus of AI tools, in particular the pursuit of neoepitopes. So these are peptides that are displayed on MHC alleles on cancer cells that you don't see in healthy cells. And already AI has been used quite a lot in trying to identify those neoepitopes, because they're notoriously challenging. And it's very important that you're confident that that really is specific to your cancer cells, so that you don't introduce a drug that, like CAR Ts, that then go and attack healthy cells as well. So I think, identifying neoepitopes, I think identifying the underlying sort of, genetic or genomic aberrations that lead to cancer. And you're seeing a lot of this work already in big pharma. I think, again, going back to in silico, a company you can tell I like quite a bit, they recently had a collaboration with a Chinese pharmaceutical company whose name escapes me, where they were looking for a novel cancer target, and they found a target and designed a drug in something like 40 days using AI. Of course, you still have to go through the whole gamut, the 2 years of development, testing, in vivo, in vitro assays, all of that. But yeah, it's not just immunology. It's being applied everywhere in biomedicine.
I think your case describes that perfect case of AI, right? Which was humans, we can see a pattern, but good God, figuring out the meaning of that pattern in large data is like, hey, I see a pattern. There's something called SNPs. Hey, SNPs are important in cancer. You could do it without AI and spend your entire life, or the robot can do it in 5 minutes going, with these SNPs and all this data, which ones are important. It gets the correlation for you much quicker, and I think that's that tool that's exciting in cancer and in my field in microbiome and in immunology, all types of stuff, to just get to the end once you have that idea in the beginning much, much faster.
Why does AI make up stuff sometimes? Because I've heard some stories of people, and maybe specific to the free version of ChatGPT, where they're asking for references for a certain question, and it actually made up answers.
AI has no idea what it's saying. It's not an intelligent agent. It has no ability to fact check itself. All it's doing, all ChatGPT does is, give it its query, and then it goes one word at a time, and it finds a word that it believes most plausibly completes the sentence that is being generated, beginning with your prompt. So, if the paper that most plausibly completes the prompt of who's the most awesome woman CEO in biotech, it refers to a paper on, Joe Viney's contributions to her amazing 30-year career, that paper might not exist, but it plausibly completes that sentence. And the AI has no fact-checking ability. So that's the problem of hallucination. And that's why I believe that, and this is endemic to large language models, it's not getting better, it's actually getting worse. And it's why I believe that artificial general intelligence is still a long ways off.
Yeah, I don't know that I would say that it's making it up. It's just used the wrong source of information and got confused in the order of the answer. So I think that's to remember that it doesn't really think, and it really is just looking for pattern recognition. And I think It's not really any different than when we see, two articles published that have differing results and you have to use your human judgment. It's not that one is right and one is wrong. Maybe one used congenital knockout mice and the other used pharmacological intervention in something. They're both true, and then they got different data sets. It's not that one's made up. It's just the way it was approached was different.
To turn this into non-math, the way I like to think about it is, help me here, Ryan, is that it's weighing a bunch of probabilities of stuff coming in and out. And sometimes we all know this in our life, the most probable things all combined is actually what happens. Mm-hmm. So it's going to spit out the most probable thing, even if that's not actually the thing.
That's right. Yeah.
Hi. So as someone who's considering a career in science writing, how do you think that AI is going to shape the way we communicate science? Whether that's through writing, making posters, visuals.
I was discussing this with the editor of Science Immunology yesterday. And so what do you think? Where is AI kind of stepping into your field? And I think on the one hand, when it comes to science, writing, and communication, first thing is that it helps you improve your output. So it helps you generate the text in a way that you have to do less of the initial work. Especially for non-native speakers like myself, it can be a huge help to help to improve your English in this case. If you're going to communicate in English, your use of the language can be very useful. On the other hand, more in the science, kind of the articles and science, scientific journals, and how the editors are using AI to improve their work and the quality of their work. On the one hand, you have people generating text with AI, which is kind of plagiarism, so you don't want that. But you can use these tools to detect it. And also you can use AI tools to, for example, pick up issues with figures, with images, image manipulation. So what you used to need to have, 5 assistants looking at this, or Elizabeth Bick, going in a crusade looking at each of these pictures. Now you can have a software that does it for you and really improves streamlines your work. And on a more, I think, more creative way, it can help you. You can discuss with AI. You can kind of bounce ideas. And nowadays, many of these language models will actually give you answers that are useful. So having a friend instead of having a colleague, you can have a pilot, a co-pilot that helps you kind of gauge whether the way that you are going about this makes sense. And I think I thought that was very, very interesting. So basically help you kind of edit your work, find the right structure for your work, look for plagiarism, look for irregularities in figures, and also sometimes maybe look for reviewers. There's also nowadays some software that helps you do that in case you're looking for people to review the papers that come to your desk, things like that. So I think there's a lot to do, or what I hear from the field, that there's a lot of options there. I don't know if you guys have anything else to add.
Well, yeah, I would say that AI is very good at looking for subtext because it's trained on so much information. You can kind of tell if maybe your logic is not quite as airtight as it could be. So asking the AI, what are some logical pitfalls that might be? What am I missing in what I've done so far? Where I think it won't help you is, often I find when I'm stuck in writing, it's not because I don't know how to write what I want to write, it's because I don't know what I want to say. And I actually don't think AI is useful for that kind of idea genesis at all yet. And that'll be the future with AGI. So I think in that case, going back, rather than trying to get ChatGPT to come up with the killer sort of next section for you, going back, thinking about what's the point you're trying to get across, and then coming back and saying, okay, here are 3 bullet points that I have of ideas. Could you flesh this out into a paragraph, an introductory paragraph? And then it'll sort of fill that in, and that'll probably help you generate more ideas and more clarity in the direction you want to go. So, really thinking about it iteratively rather than as producing output.
With science communication, though, one of the big things is the communicator, and it creates a network, right? So people listen to podcasts or go to blogs based on their experience there before with the personality of people doing it. And so while these large language models can help write something that sounds pretty good because it's based on the sum total of human writing, it's not a person's writing or that person's style. And so in so much is science communication, there's that personal element, which is a commodity, right? It's the value. I like listening to this person on the radio or reading this person's style. That can never be supplanted by anything until you have an AGI, which has its own style. And so I think that's also really important to know if you're looking at careers in science communication and writing, that the personal element's actually paramount.
Yeah. Who would not want to have the personality behind the voice, right? Or the pen or the text.
Take us out of here, Brenda.
All right. So I think with that being said, it's been such a wonderful conversation. I thank our guests so much, Joe Viney and Ryan Peckner. Thank you for coming and talking to us. And thank you, all of you, for also being here, for sticking to it. And as always, if you have any want to hear more about the podcast, we have a website, www.immunologypodcast.com. You can find us also on Twitter, X, at Immunopodcast. And if you have a question or you want to suggest a guest, so we are always happy to hear from you, the listeners. So please reach out if you feel like.